Yeah, next topic is flow injection analysis and sequential injection. Okay, um, there are two types of analysis. One is flow injection, and the other one is a batch process. Okay, batch process is often uh, used in analysis, which is that individual samples are separately analyzed. Okay, let's let's think. You have one set of sample, and then you do the analysis for the sample. Okay, separately, and then after you finish the analysis, and then you do the another analysis for the next sample. Okay, yeah, that kind of analysis is batch processes. Okay, but in flow analysis, flow injection analysis means you you have continuous flow, mm, and then you inject your sample here. Yeah? Injection after injection. Okay, that's the continuous mode. The advantage of this flow injection mode is speed, so you can analyze your sample more rapidly, right? And then automation of sample handling is possible, and this is low cost. Okay. In this flow injection analysis, a liquid sample is injected into a continuously flowing liquid carrier. Okay, now this you need much carrier liquid, and it's continuously moving by this peristaltic pump through the injection valve and through this mixing coil, and eventually through this part. This is flow cell for spectrophotometric detection. Okay, and then uh, you add the reagents into this carrier flow. So actually this carrier flow include contains reagents. Why? Because this reagent should react with the sample. Okay, you will see the example in the next slide. Okay, a sample flows from injector to the detector. Okay, sample zone broadens here when immediately after you inject your sample into this injection valve, and then samples are confined in this small area, but now it is carried away by this carrier flow, right? And then it moves down through this mixing coil. Okay, and then its sample becomes more and more broadened. All right. Yeah. Okay, this is actual photograph of a mixing coil. Okay, so you inject here, and then now it pass through this enters this mixing coil here, and it is a rather long coil. So inside the mixed coil, your um, sample and reagent now mixed, and they react to give rise to a colored product. Okay, here is an example of flow injection analysis. The determination of herbicide acetochlor in food. Yeah, this is herbicide that should not be in food. To analyze this herbicide, first you hydrolyze this one at elevated temperature at 100 degrees C in zero point 0.2 molar hydrochloric acid, and then it is hydrolyzed, and then this one is the hydrolysis product, but it is not colored. Okay, so you need another um, treatment, yeah, which is the reaction with reagent. Okay, now you inject. Okay, this hydrolysis product is injected in the carrier stream. Yeah? So inside the carrier stream, which contains the reagent, yeah, this is a reagent, thiazonium, uh, this reagent in a carrier stream, and now reacts with this hydrolysis product. Okay, and then this one. The product is colored, right? So since it is colored, so it can be detected spectrophotometrically. Okay. So here, the carrier stream contains a diazoxide salt, which is the reagent that reacts with the sample to give a colored product, which can be measured at a visible absorbance at 400 nanometers. Okay. Yeah. So this figure is illustration how the samples are broadened inside um, the reaction tube. Okay, this is the tube. This is a helical coil, right? And carrier flow is always flowing from the left side to the right side, and then here you inject your sample, right? At first, your sample is confined only in this area, right? But this front end is curved this way because carry the flow now carries away your sample. But now think about that. This one, this tube is filled with carrier flow plus reagent, okay? And then here reagents react with the sample, yeah? All at once? No. In the front end here, and then also um, this end, okay, this back end, this part, and then inside the middle part of the sample is not immediately reacted, okay, but as the carrier flow carries away this sample, and then slowly this inside the sample also reacts with reagent. Okay, but this one is the zone rich in unreacted sample. Okay, this is illustration of how the sample reacts with reagent. All right. Curved leading and tailing edges. Okay, this is uh, the leading and tailing edges are curved. Okay, liquid near the walls mixes with liquid in bulk by radial diffusion. Okay, here radial diffusion is what? Through this one, this way, not this way. Okay, this is radial diffusion. Okay, this is radial diffusion is perpendicular to axial direction. Okay, so liquid mixes first near the wall. Okay, near the wall, but not immediately inside the sample. The narrower tube, the faster is the radial mixing. Yeah, it's natural, right? If this thickness is narrower, so this radial mixing must be much faster. Okay, mixing occurs in a helical mixing coil. Okay, here, this is helical mixing coil, or it is also called the reaction coil because the reaction occurs inside that coil. Curvature and sharp bends in the flow path create turbulence which promotes mixing. Why this coil is has this helical shape? Because it is to save this distance, and also this one is curved, and whenever the flow you know the this carrier flow goes through this one, yeah, this helical coil, and then mixing becomes much easier. Okay, so curvature and sharp bends in the flow path creates turbulence which promotes mixing. The key to analytical precision is the repeatability of a flow injection. Okay, the flow injection repeatability is really important. So every time you inject the same amount of the sample, you should have the same result within certain experimental error. Okay, the concentration profile of a product passing through the detector depends on many conditions. Okay, including volume of a sample, flow rate, reaction rate, and temperature. Okay. The detection flow cell has a G-shaped liquid path here. It has a G shape. Okay, now flow after leaving this helical coil, this flow now enters this G shape, you know, uh, this path. Okay, liquid path. Monochromatic light is brought by way of a fiber optic. Okay, light that has passed through the flow cell travels to the detector via another fiber optic. So the spectrophotometric detection of the early light becomes possible. All right. Yeah, this is another example with more complicated structure. Basic structure is the same, but here um, it has two more paths. Before a picture, there is only one, path. Okay, and this this line, this carrier liquid has reagent. But what if you need two reagents? Okay, reagent 1 and 2. In that case, you have to have two um, lines of injection. Okay, for reagent 1 and for reagent 2 here. Okay, look at this picture. This part is only difference. Okay, here is an example: ammonia analysis and seawater. Okay, in the case you need two reagents. One reagent is ortho butylaldehyde, and the other reagent is sodium sulfite plus formaldehyde. Okay, you um, input reagent 1 through this line and reagent 2 through this line, and after there's a sample injection, sample and reagent 1 and reagent 2 are mixed here. Okay, all three samples are mixed, and then they pass through this helical coil. Now it is maintained at 65 degrees C. Okay, and the reaction takes place in this helical coil. Then it moves to this um, fluorescence detector cell. Okay, the fiber optic. Okay, here there is exaggeration, and flow in and flow out. This is a light excitation light enters in through this one, and then emission line. Okay, this emission line is perpendicular to the flow direction. Okay, here you detect the fluorescence. All right, in the detector the solution is irradiated with 365 nanometer ultraviolet light. Okay, in this case you detect not absorption but fluorescence. Okay, so you excite the sample by 365 nanometer, which is ultraviolet light, and then emission occurs at 423 nanometers. Okay, you need this fiber optic and another fiber optic. Okay, but they are placed at right angle. Okay, at a right angle. Yeah, this figure shows the repeatability of the injection. As I said, in flow injection analysis, repeatability is really important. Every time you inject the same amount of same concentration of a sample, and you have to get the same result. This is the response to replicate injections of ammonium chloride standard. Okay, here you inject 100 nanomolar solution and 200 nanomolar solution and 300, 400. You see, almost identical result was obtained, but not exactly identical. There is some uh, standard deviations around, for example, uh, for 200 nanomolar samples, about 2.2% standard deviation, yeah? And for 1 nanomolar solution, more than 6% of standard deviation. But this kind of repeatability is actually good. Okay? So with flow injection, typically you can measure 100 samples per hour. Okay? At the speed is I think is enough, right? Now let's learn the another types of analysis, which is called sequential injection. It is distinguished from flow injection by flow programming and flow reversal under computer control. Right? This is sequential injection is more advanced injection flow injection analysis, but it is a little bit different from this flow injection in that it can be programmed and controlled by a computer, and the flow direction can be reversed. Okay, in ordinary flow injection analysis, flow direction is only one direction. Okay, only one way. But in this sequential injection, flow direction can be reversed. So there are many features. Flow is not continuous. Okay, in this case, flow is not continuous. In flow injection analysis, one of the disadvantages is that you need much carrier liquid. Okay, because you have to have a continuous flow, right? So it consumes a lot of your carrier liquid. But in sequential injection, you don't need to have continuous flow. Okay, so smaller volumes of reagents are required and less waste is generated than in flow injection. Okay, since you don't need to have continuous flow, so you need only a small volume and is required, and so the waste generation is less than in flow injection analysis. Okay, and sequential injection has been miniaturized to the point where it is also called a lab on a valve. Okay, one of the advantages is miniaturization, so you can uh, make it very small. Okay, so sometimes it is called lab on a valve. One lab is on a very small valve, right? That's the concept of lab on a valve. Okay, if one lab is on a chip, that is called lab on a chip. Okay, and miniaturization and discontinuous flow reduce the consumption of expensive reagent. Yeah, this is quite an advantage. You need sometimes very expensive reagents such as enzymes and antibodies for biochemical assays. Yeah. This enzyme and antibodies are usually very expensive. So for the flow injection mode analysis, you need a lot of enzymes and antibodies. That is, you have to spend a lot of money, right? Nobody wants that. And sequential injection is used for continuous monitoring of environmental and industrial processes, even at remote locations. Okay, yeah, this is application. Yeah, this is a basic structure of flow injection analysis. The central feature. Oh no, I'm sorry. The sequential injection analysis. The central feature of the sequential injection apparatus is a six-way valve. This central figure is the six-way valve. It has one, two, three, four, five, six ports, right? And then this central port now it turns, right? This one. If the central port now connected to port 5, flow from here to C to 5 and to waste. Okay, if this arm now turns to port 2, and then flow direction changes C to 2. Okay, when it is connected to 3 and the C to 3. Yeah, like that. So in sequential injection analysis, this six-way valve is very important. That this is the central part. Okay? This figure shows a connection between port 5 and central port C. So liquid samples are brought to port 5 by auxiliary peristaltic pump here, or you can flow your sample from here, yeah, using peristaltic pump here through the port 5 to port C. Okay, the flow direction can be reversed. Okay, near the upper left here is a stepper motor controlled syringe pump. This is syringe pump. This syringe pump, you see, you push this piston, yeah, at a very precisely controlled speed. The syringe pump, which moves precise volumes of liquid forward and backward. So you can move the syringe pump forward and backward either way. So if you move forward and then flow direction is like that, this way. If you move backward, the flow direction is from here to there, right? Backward. The valve at the top of the syringe pump, this is another valve. This is not six-way valve. Maybe this is, I think, three-way valve, right? One, two, three. This one connects the syringe to the reservoir or to the this holding coil. Okay, now it shows that this arm is connected to this one. Okay, this one is connected to this holding coil, and if this part is connected to the carrier buffer, okay, and then flow direction becomes different. All right, and now here you inject. This is your sample. Now you inject a sample through this one, yeah, and then also you inject reagent 1 or 2 by simply changing this arm direction. Okay, after you inject for sample, and then now you change this arm to reagent 1 or 2. Okay, that way you can inject your reagent into this holding coil where your samples and your reagent react together, okay, and mixed here. Okay, and this one, and after mixing here, and then you inject using this syringe pump, you inject this carrier buffer through this part, and then now this part, and then the central arm now connected to this port 2, and then this one. After reaction here, and then your sample is carried to this flow cell and detected by spectrophotometry. All right. Yeah. So in this case, you don't need a continuous flow. After you inject a certain amount of your sample or reagent 1 or 2, and then that's it. Okay, that's it. You don't need to keep flowing your reagent. Okay, you can precisely control the injection amount with this sequential flow. Why sequential injection? Yeah, sequential means first the sample injection and reagent injection. Okay, and that's why it is called sequential. Yeah, this figure is illustration of the injection. Yeah? And first your sample injected. This is holding coil, and then now sample moves this way, and then you inject the reagents, this one, and then while the liquid are moving toward the left side, then here the sample and inject reagent react together, and you stop the flow and then reverse the flow direction. Okay, and then here the direction flow direction is to the right side. That way the sample and the reagent mix more completely, and now it goes through to the detector. All right, so the main feature is that the six-way valve is needed, and flow a certain amount of the sample is only required. Okay, so you don't need to have a continuous flow, and also and the flow direction could be reversed. All right, that's the main feature. Yeah, this is actual figure of sequential injection apparatus. Okay, this part is six-way valve, and this is the this fiber optical fiber connected to spectrophotometer, and this is a holding coil, right? And inside this coil, the reaction uh, completes. All right. Yeah.